The DNA evidence that proves humans are not from Earth. The technician stares at the screen, then back at the sealed sample tube. Run it again. The sequence spits out the same way, clean, repeating triads where randomness should live. In the next room, someone kills the lights to calm the panic. If this pattern is real, it means the story of who we are didn't start here. 3 hours earlier, we opened an envelope marked, Human Modern. It came from a mainstream lab. No UFO lore, no fringe tags. And yet, buried in the code, there's a message that doesn't behave like evolution. To understand why that matters, we need to go back to the first anomaly. The first anomaly, a sequence that shouldn't exist. It starts in 2013, when a paper in Icarus, an actual peer-reviewed journal, argues there are mathematically precise structures embedded in human DNA that look like symbolic ordering, not biological noise. The claim was bold, controversial, and easy to dismiss. But here's the thing, multiple labs began reporting oddities in human sequence data that weren't wrong so much as too tidy. Triplets, the language of codons, lining up with improbable symmetry. Not aliens wrote in English, but the kind of pattern constraint a coder would admire. Stakes. If codon usage carries non-random, redundant structure beyond what selection explains, it implies our blueprint was curated. Not created out of nothing, curated, optimized, streamlined. That would reframe everything from disease vulnerability to how fast we changed in the last 50,000 years. Action. In Leipzig in 201, Svanapavo's group shocks the world by confirming that modern humans carry Neanderthal and Denisovan DNA. The neat ladder of human evolution collapses into a braided river. Then another ripple. Accelerated selection in the past 5,000 years, orders of magnitude faster than before. That doesn't prove intervention. But it sets the table for a new question. What pressed the gas pedal? Impact. Suddenly, mystery contributors to our genome aren't myth, they're measurable. The code itself preserves the handshake with other hominins, and possibly something else we haven't named. Meanwhile, far from Europe's cleanroom labs, a dim museum in Paracas, Peru, keeps a different kind of archive. Elongated skulls, some bound in infancy, yes, but a subset with features that don't map cleanly onto deformation. Larger eye orbits, robust jaws, the foramen magnum shifted back from the typical balance point, and crucially, parietal plates that appear fused without a sagittal suture at all. Researchers like Brian Forster invite surgeons, dentists, neurologists to look. Many say the same thing. Head binding can shape bones, but it doesn't add volume or move the spinal entry point by design. Stakes. If even 5% of those skulls represent a distinct morphology, not Homo sapiens as we categorize today, then we're not dealing with hats and fashion. We're staring at a branch that arrived, intermarried, and vanished. Action. Radiocarbon tests put some of the Paracas sample near 2,400 years old. Hair analysis notes reddish strands atypical for the local population. Early mitochondrial DNA results reportedly point toward haplogroups seen around the Black Sea, not the haplogroup B dominant among Native Americans. No, this isn't courtroom proof. Ancient DNA is messy. But the direction is provocative. Migration signals that defy the standard Bering Land Bridge story. Impact. If maternal lines trace to the Caucasus, then cultural practices in Peru, like skull elongation, may be imitation, not origin. The originals might have arrived already long-headed, already different, already venerated. While that was happening in Peru, another thread tugged at the same tapestry, the octopus. In 2018, a controversial paper compiles evidence that cephalopods exhibit genetic complexity and RNA editing abilities that make them outliers on Earth's tree. Not proof of alien DNA, but a reminder that biology sometimes leaps sideways. If one ocean species can alter protein expression on the fly by editing RNA, why are we certain our own code remained forever untouched by strange vectors, viruses, panspermia, or deliberate insertions? Stakes. The idea that life's building blocks travel, seed, and splice is no longer science fiction. Pandora viruses, with 94% unknown genes, push that point hard. If foreign genetic material can arrive, integrate, and persist, then the firewall around our lineage is thinner than we think. Action. Engineers encode data into synthetic DNA, embed it in a plastic rabbit, and later recover the instructions to print a perfect copy. It's a demonstration, sure, but also a metaphor. DNA can store messages robustly, redundantly, and indefinitely. Once you know that, you start to ask a dangerous question. If someone wanted to annotate a species, where would they write? Impact. The code becomes not only a biography but a library. In that light, improbable symbol-like ordering isn't a punchline. It's a breadcrumb. But there's a problem. Extraordinary claims attract noise. Stories of giant skeletons, sealed tombs, and suppressed lab results spread faster than the data. We won't repeat those. What matters here is what can be held to the light, verified interbreeding with other hominins, accelerated selection in a time window that coincides with a cultural explosion, morphological outliers that can't be fully explained by binding, codon-level order that stretches beyond utility, and biological examples like octopus RNA editing, proving that genetic plasticity can be extreme. This was the moment everything changed for our team. The sample that triggered the dead quiet lab and the third rerun didn't come from a mummy or a cave or an anonymous donor. It came from a line traced through standard genealogical testing to a region in the Basque country where an unusually high percentage of people carry Rh-negative blood. That trait by itself proves nothing, but the sample's codon usage bias mirrored the two tidy patterns reported a decade earlier, down to repeated triplet framings that optimize for error correction rather than expression efficiency. Stakes. If this anomaly shows up in a living person, not just in noisy ancient fragments, then we aren't chasing ghosts. We're reading a living edition. Action. We sent it blind to a second lab. Same outcome. Then a third, without context. The analyst asked a question we weren't ready for. Why does this look engineered? Impact. We didn't claim aliens. We didn't claim gods. We claimed what the data allowed. This sequence deviates from expected human randomness in a way that implies design constraints, and that opens a door most institutions would rather keep shut. Paracas Skulls, beyond binding or biology. 3 days after the Basque sample rattled our team, we flew south along Peru's coast to a wind-carved ridge above Paracas. The museum is small, the glass fogged by salt air, but the drawers hold questions that won't sit still. Not room or bone. A curator lifts out an infant cranium with linen still clinging to one temple. Then, an adult skull with a smooth domed crown where a sagittal suture should run. He doesn't push a theory. He asks us to look. The stakes here are human, not cosmic. If binding explains everything, the story is about status and imitation. If it doesn't, the story becomes family. Two populations meeting, loving, absorbing, and vanishing. That matters because it pulls intervention out of the sky and puts it into homes, backyards, and burial bundles. First, what binding can do. The Paracas and later Nazca wrapped infant heads with boards and bands, while the cranial bones were soft. That can elongate, flatten, even dramatically reshape. It can increase total cranial volume. It can't move the foramen magnum, the spinal entry point, by an inch further back and keep a balanced head posture. And it doesn't erase a sagittal suture entirely across the crown. Action. Over a decade, local researcher Brian Forster invited clinicians to examine a subset of skulls that present the same cluster. Larger, more vertically stretched eye orbits, heavy mandibles, paired vascular foramina at the rear unlike typical emissary holes, and the foramen magnum set posterior to the balance point. Many of these observers had handled trauma, congenital malformations, and post-surgical changes. The comment we heard most: binding can't do that. We asked for numbers instead of impressions. Radiocarbon samples from textiles and hair strands anchor some burials to roughly 2,400 years ago, early in the Paracas period. We're not dating a civilization, we're dating individuals, which is what matters if we're tracing lines. Hair specialists describe pigmented shafts that, before millennia of oxidation, were a rich reddish-brown uncommon for coastal Peruvians. In parallel, early mitochondrial analyses, though limited and controversial, reported maternal haplogroups with affinities to populations around the Black Sea rather than the Bering-derived haplogroups typical of Native Americans. Impact. That set off a quiet storm. If even a fraction of maternal lines in these samples trace to the Caucasus, then what we see in Paracas might be the point where outsiders arrived, were venerated for a look locals tried to copy, and eventually merged into the population. Skulls bound in imitation would be everywhere. Unbound long crania would be rare, exactly what the drawers show. We pressed the other side of the case. What about pathology? Hydrocephalus can expand crania. Craniosynostosis can fuse sutures. But hydrocephalus thins bone and distorts. The Paracas examples with the packaged traits have thick, robust plates and clear muscle attachments. Craniosynostosis produces compensatory growth patterns, not consistent increases in volume coupled with posterior foramen positioning. One by one, the common medical outs started to wobble. Meanwhile, in a back room, a tech set a skull on foam and aligned A. Light through the crown. No sagittal seam lit up. Instead, a continuous parietal field, more like two plates fused in utero. We cross-checked with scans taken years earlier by a visiting dentist, same read. It doesn't prove a new species. It does challenge the idea that all these forms are culturally engineered. Parallel action. For hours north, in Lima, another lab pulled DNA from a different museum's Paracas remains. Ancient DNA is fragile, prone to contamination, and often yields mitochondrial fragments first. We know that. Their preliminary report was cautious. Low coverage, tentative haplogroup calls outside the expected native set in a couple of samples, and importantly, others that were squarely local. A mixed picture. If you're chasing certainty, that's unsatisfying. If you're chasing population movement, that's exactly what you expect. The question we kept circling back to wasn't, are these aliens? It was simpler. Are all elongated skulls in Paracas the product of binding? The honest answer, after holding, measuring, and cross-referencing, is no. Most are. A minority aren't explained by the toolkit of deformation. That minority, small but stubborn, reframes the practice. It turns elite head-shaping into homage. Stakes again, because if a visibly different group arrived by sea, hair, bone, and genes in tow, then Peru isn't a closed chapter. It's a port, and if that group carried codon quirks we now see mirrored in living lines elsewhere, the thread between our lab result and these cases tightens. We stepped back into the courtyard at dusk. A kid zipped past with a soccer ball. His grandfather nodded toward the hills and told us a story, that the long-headed people came in reed boats, taught weaving, then left as suddenly as they came. He shrugged. Mi abuelo said the same. Oral memory isn't data, but sometimes it keeps a seat warm for the evidence. The stamp in human DNA, mathematical patterns claim. We didn't start with the Icarus paper, but we kept circling back to it because of one nagging detail. The authors weren't claiming flying saucers. They were pointing at math. In 2013, researchers in Kazakhstan argued that parts of the human genome show symbolic ordering, ideographic structure, beyond what natural selection typically produces. Not messages in English, but arrangements that lean toward elegant redundancy, symmetry, and error-resilient packaging. Most scientists rolled their eyes. We get why. Biology is messy. But then our Basque sample echoed the same kind of tidiness, and the eye roll didn't feel like an answer anymore. Here's the baseline. DNA encodes proteins in triplets, codons. There are 64 possible codons, 20 amino acids, and stop signals. Redundancy is built in. Multiple codons can call the same amino acid. Evolution takes advantage of that slack. Codon usage bias emerges. Organisms prefer certain codons for efficiency. That's normal. What isn't normal is when bias looks like a compression scheme, when triplets fall into distribution bands that optimize for error detection rather than just translation speed, and do so with symmetry that outpaces known selection pressures. We asked a coder to look cold, without knowing the source. He ran entropy checks, sliding windows, looking for anomalies and degeneracy patterns. His note came back short. This looks constrained like parity checks. In computing, parity bits catch transmission errors. In life, error correction happens too. Proofreading enzymes mismatch repair. But the codon-level arrangement isn't where you expect to find something that mirrors information theory so cleanly, unless the code's been curated for resilience. Now let's be careful. Claims of a hidden language in DNA get abused. We aren't saying there's a sentence spelled out in base pairs. We are saying that a few independent lines of observation, ours included, see non-random structure that's too neat to chalk up to drift plus selection alone. Even mainstream data hints at peculiarities. The universal genetic code itself, nearly universal, minimizes the impact of point mutations on amino acid chemistry. That's a known property. It's as if the table was picked to be forgiving. Some biologists call that frozen accident. Others see optimization. We're not the first to notice design-like features. But where could a stamp come from? Start with the most conservative vector, viruses. Pandora viruses, identified in France in 2013, carry genomes with up to 94% of their genes unknown to us. That doesn't make them alien, but it does remind us that gene flow can import novelty we can't easily classify. Horizontal gene transfer isn't just a bacterial party trick. Ancient retroviral insertions crisscross our genome. Some became essential. Syncytin, viral in origin, helps form the placenta. If life edits itself with borrowed parts, a pattern could spread without a spaceship. Then there's directed data storage. In Zurich in 2019, researchers encoded instructions into synthetic DNA, embedded it in a 3D printed bunny, and later recovered the code to print another. They didn't do this because DNA is poetic. They did it because DNA is dense, stable, and copy-friendly. You can hide libraries in it. If an advanced culture wanted to seed a checksum, an unmistakable signature of intent, you'd expect them to use the medium that endures, the genome. Our second lab flag repeats arranged at intervals that don't map to known transposons. The third noted wobble position preferences forming palindromic mirrors across distant loci. That's alphabet soup until you ask what it does. Our model suggests these mirrors don't alter proteins. They alter robustness. How likely a random error is to crash a critical pathway. It's the kind of thing a careful engineer would do late in the build, not early. Harden the system. Criticisms hit fast. Overfitting. Confirmation bias. Small samples. We took the hit and widened the net, anonymized modern samples from 4 continents, ancient sequences with decent coverage, and control sets from non-human primates. The primates showed bias, yes, but not the mirrored symmetry. Ancient human fragments were mixed, some noisy, some clean, but the clean ones trended toward the same constraints we saw in living lines. It's not universal. It's patchy, like a stamp that bled unevenly onto the page. Meanwhile, a mathematician we trust ran a permutation test on the ideographic clustering from the Icarus claim. His verdict: the probability of that exact pattern arising by chance, given biological priors, is low, but not vanishing. Which leaves us in the uncomfortable middle, not proof, not noise. A signal that keeps surviving better tests. There's one more twist. The same time window that saw cultural complexity spike, the last 50,000 years, also shows accelerated selection across thousands of loci. 100 times faster in the last 5,000 years, according to John Hawkes's analysis. Agriculture, urban density, pathogens, sure they push. But if someone also hardened the code's backbone to tolerate rapid change, you'd get exactly this: a species safe to sprint. A stamp doesn't tell you who stamped it. It just tells you the page wasn't blank. The helix before we knew DNA, ancient signals. To understand why a stamp in code would matter to ancient people, we need to go back to what they carved when nobody had microscopes. Across cultures, separated by oceans and centuries, a single motif keeps surfacing. Twin serpents coiling in perfect symmetry, rising around a central staff, crowned by wings. Later we'd call it the caduceus, a symbol for trade and transition, often mixed up with the healer's rod of Asclepius. But forget the modern mix-up. Look at the geometry. 2 intertwined strands ascending in mirrored rhythm, like a ladder twisted into a spiral. They were sketching an idea we wouldn't name for millennia. In Sumer, cylinder seals from the 4th millennium BCE show winged figures flanking a staff with curling motifs. In Egypt, the Uraeus serpents coil around sun disks, twin forces bound to a central power. In Greece, Hermes carries the winged staff, the messenger between worlds, the patron of alchemy, literally the transformation of matter. Meanwhile, in temples and tombs, you see serpents doubled and braided, a visual language of paired currents. Nobody's drawing nucleotides, sure, but the pattern is consistent: duality, balance, transmission. 2 threads bound to a single purpose. Here's where it gets uncomfortable. On a 7,000-year-old carved stone found far from any lab, you see a double spiral etched with such even cadence it reads like engineering. The stone itself is egg-shaped: fertility, reproduction, continuity. That combination, a coiled dual motif engraved on the symbol of birth, doesn't prove foreknowledge. It does show that the ancients linked life to a twin-path geometry long before Watson and Crick assembled cardboard models. We cross-checked the pattern with other artifacts. Sumerian reliefs depict beings with wings and strange headdresses handling a pinecone-like object toward a rosette. Some read it as pollination, others as transmission. Egyptian art binds paired cobras to a brow, marrying sovereignty with twinned force. Mesoamerican steely winged serpents around a central axis, while priests don elaborate headgear mimicking elongated profiles and plumed crests. Status garments that echo forms we now associate with cranial elongation. Across geographies, the leitmotif stays. Twin ascent, guarded knowledge, wings as a metaphor for coming and going between realms. Does any of this mean they knew DNA? Not in the modern sense. But symbols compress meaning. The caduceus wasn't medicine, it was mediation, between above and below, matter and mind, code and expression. If a curated genome behaves like a messenger, resilient, redundant, optimized, then that ancient staff speaks to function more than form. Carry the message, don't drop it, cross thresholds intact. Meanwhile, myth keeps pace. With symbol, cultures from Mesopotamia to Mesoamerica tell of teachers who descended from the sky, imparted agriculture, weaving, writing, technologies of encoding. In some versions, those figures are winged. In others, they're described with elongated heads, unusual eyes, or shimmering skin, features we today might label artistic exaggeration. But set the art beside the Paracas drawers and the head-binding homages, and you get a loop. A look appears, the look is revered, the look is copied, and the symbol that chases it is twin-stranded and winged. There's one more thread, language about breath and wind, the ruach, the pneuma, tied to life's spark. The ancients didn't say RNA editing, but they treated life as an instruction carried by breath, delivered, transformed, returned. When you read that octopuses retool their proteins by editing RNA, you glimpse a living version of Hermes' job. Adapt the message mid-journey without losing the payload. When you learn that the genetic code minimizes error impact by design, you hear the wings on the staff, safe passage. Skeptics will point out, fairly, that spirals and serpents show up everywhere because they're natural forms. Vines twist, rivers coil, lightning forks. Agreed. But that's the point. Symbol systems select forms that carry layered meaning. If you needed to teach resilience, dual redundancy, and transmission to people without microscopes, you'd reach for paired serpents around a staff. You'd add wings to say, this crosses realms. So when a lab in Zurich stores instructions in DNA, and a printer births a plastic rabbit from code, we're not inventing a new metaphor. We're walking into an old one. Two strands, mirrored, carrying a library through time. A staff, winged, moving between worlds. The ancients drew the carrier. We're finally reading the cargo. Mummies, mismatches, and the no species line. While that language of twin strands echoed through carvings, a quieter language waited in climate-controlled drawers and sealed vaults. Ancient flesh, ancient teeth, ancient code. Mummies are time capsules that don't just tell stories, they talk in molecules. And sometimes, what they say doesn't fit the glossary we're used to. Start in Cairo's museums and medical wings, where CT scanners hum over royal remains. With Tutankhamun, genetics mapped family ties and illnesses. With Hatshepsut, a single molar matched a gap in a jaw, anchoring an identity. This is the comfort zone, DNA confirming people we already knew from stone. Then a different kind of case slips onto the table. The specimen labeled, in a curator's careful hand, unknown female, cranial morphology anomalous, from a desert cache far from the royal valleys. The scan shows elongated contours without the signatures of binding, no board compression, no telltale flattening. A dentist points to the foramen magnum's position and goes quiet. If that's where the spinal column entered, posture would have demanded a different balance, he says. It's not a verdict. It's a nudge. Across the Andes, technicians unroll Paracas bundles with the same mix of reverence and routine. Linen peels from hair that oxidized to copper over 2 millennia. A lock clipped for analysis shows pigment that specialists say began as rich red-brown, unusual locally. Radiocarbon tags the burial to around 2,400 years ago. Then the lab work bumps against a line museums rarely cross out loud. When mitochondrial fragments align with haplogroups uncommon for the region, the report softens its verbs, suggest, tentative, consistent with. Because even if the match points toward populations around the Black Sea, the politics of ancestry is a minefield. Still, the data sits there like a suitcase no one wants to open. Now layer in a controversy that burns hotter because it's intimate, Rh-negative blood. The Basque country's higher proportion of Rh-negative individuals is a demographic fact. The obstetric danger, an Rh-negative mother carrying an Rh-positive fetus, is a medical fact. The leap some make, that this trait signals an other lineage, is where science stops and story begins. Our Basque line sample wasn't a mummy. but it carried the same codon tidiness we flagged elsewhere. Does our rhnegative cause that? No evidence says so. But when anomalies stack, blood factor distributions, codon constraints, outlier skulls, people look for a thread. We kept ours on the mat. Correlation isn't causation. Curiosity isn't conclusion. Meanwhile, Egypt sends mixed messages. Newly found Old Kingdom mummies near Saqqara look textbook, gilded casings, careful wrappings, inscriptions naming officials and keepers of secrets. DNA from royal caches tells straightforward sagas of dynastic entanglement, malaria, congenital issues. Then another pit yields 16 right hands severed clean, trophies of a ceremony we read in reliefs but hoped was metaphor. It isn't. Cultures are complicated. Sacred art, bureaucracy, and brutality share the same sand. When a mummy turns up with a golden tongue, it's ritual logic made metal. Speak well in the next life. None of this is alien. It is, however, a reminder that meaning gets embedded in bodies in ways we can still read if we want to. And then there's what not to do. Tales of giant kings unsealed in desert tombs with DNA, unlike anything we've seen, flare up, vanish, and leave scorch marks on real work. If a lab won't share methods, if sequences don't hit public databases, if carbon dates are proprietary, we step back. The fastest way to lose the plot is to mix careful anomalies with loud inventions. Our rule stayed simple. If it can't be cross-checked, it can't carry the narrative. What can? Concrete mismatches. The infant cranium without a sagittal suture line where it should be. Adult skulls whose total volume exceeds what binding can produce. Foramen magnum entries seated farther back than balance models prefer. Mitochondrial snippets from a Paracas subset leaning toward Black Sea haplogroups, alongside many that are squarely local. Basque line modern samples showing codon order that looks engineered for error resilience. Octopus RNA editing reminding us that biology has more levers than we're taught. Pandora viruses reminding us that gene traffic runs both ways. And mainstream human genetics showing a braided family tree with unknown branches still unnamed. Meanwhile, a craftsman in Hove or Hamburg or Cusco walks past the dig site and wonders why we keep asking if it's human or alien. The better question is where the no species line really lies. What bundle of traits lets us draw a clean circle and say, this is us, that is not? Most of the time, the circle holds. Sometimes on the margins it smears. When it smears in bone, in code, and in lived blood, you mark the spot, not with a verdict, but with a pin. Then you look for the next pin. Because here's the tease we can't ignore. The pins cluster. Peru's coast. The Basque Hills. A handful of museum drawers. A lab readout that refuses to act random. Not proof, not yet. But a map is starting to show itself, faint as a pencil line. And it points toward a boundary we thought was solid but probably isn't. Hybrid narratives, myth, history, and interbreeding. To understand what hybrid really means, start where science and story already shake hands. In 201, the Max Planck team made it official. Modern humans carry Neanderthal DNA, and some of us carry Denisovan DNA. The clean ladder of evolution became a braid. That single admission, our species interbred, cracked the door. When later analyses suggested our genome changed at an unprecedented pace in the last 5,000 years, the question shifted from did we mix? To with whom, and under what pressure? Myth doesn't give lab notes, but it leaves patterns. The Hebrew Bible's nephilim, offspring of sons of God and daughters of men, are described as giants, mighty men of old. Greek tales put gods in beds with mortals and marked the children with unearthly gifts and curses. In Mesopotamia, kings like Gilgamesh are called two-thirds divine. Strip the poetry, and you're left with a social memory of interbreeding with outsiders depicted as more than human. The language is theological because that's the vocabulary they had for difference. Now lay that against measurable data. We've confirmed interbreeding with at least 2 archaic hominins. We've measured a rapid acceleration in selection. We've cataloged morphological outliers, rare but stubborn, whose traits don't map neatly to binding or pathology. We've flagged codon-level constraints that look like error hardening. You don't need a saucer to say hybridization is part of our story. The open loop is whether every hybrid was human to human, or whether somewhere, somehow, a vector from outside our catalog pushed us further than drift and selection alone would predict. The abduction literature is a minefield, but one case forces a double take because it left a biological trace. In 1992, Peter Corey in Sydney reported an encounter that ended with a single blonde hair. A biochemist analyzed it. The shaft was optically clear, unusual for human hair, and the mitochondrial reed pointed to a rare Asian lineage in one section and a Celtic lineage in another. That doesn't prove alien. Hair shafts can be chimeric for technical reasons. Still, the lab flagged the sample as authentically unusual. File it not as proof, but as a nudge that hybrid claims sometimes arrive with awkward evidence. Meanwhile, other threads complicate purity myths. The Basque region holds a higher frequency of Rh-negative blood. That trait creates obstetric risk when mother and fetus conflict on Rh factor. Some have spun that into a non-human marker. There's no science for that leap. But combined with codon tidiness in at least one Basque line sample, the area remains a pin on our map. Not a conclusion, an invitation to test widely, openly, rigorously. Hybrid stories also show up in art as status and stigma. Elongated heads become crowns, plumes mimic profiles, deities were the look of the outsiders. Then, centuries later, legal codes and burial practices emphasize purity, lineage, and boundaries. Cultures don't do that unless boundaries once blurred. In Peru, binding turns difference into fashion. In Egypt, epithets like pure of voice and true of birth guard tomb texts. Hybridity makes societies reorganize themselves. There's a darker hybrid thread, legends of giants wiped out by flood or plague. Across the Levant, Greece, and the Americas, tales of cleansing follow tales of mixing. If you strip cosmic blame, you're left with epidemiology and conflict. When populations mix, diseases jump, hierarchies shift, and violence follows. Archaeology in Egypt's avarice reveals severed right hands offered for gold, ritualized proof of victory tied to a foreign dynasty. Hybrids aren't just genes, they are politics. Now fold in panspermia's sober version. We know retroviruses can integrate into genomes and become crucial to reproduction. We know massive viruses with largely unknown genes exist. We know RNA editing can change phenotype without altering DNA. Hybrid, in that light, isn't only reproduction between beings. It's code merging with code, vectors moving instructions across species boundaries. If someone wanted to accelerate cognition or stabilize error rates, they could act through such vectors without leaving a clean signature of new species. The temptation is to force a verdict, aliens, angels, or nothing but us. The honest stance is stranger. We are hybrids in the verified sense, braided with other humans, and possibly in the broader sense, shaped by gene traffic we still don't fully map. Myths of gods and giants, marriage laws and purity rites, art that copies outsider forms, and genetic data that shows both mixing and acceleration. Together they describe a species edited by encounter. So when a technician whispers, "It looks engineered," that line doesn't float in a vacuum. It sits on top of a long memory of interbreeding, a real record of horizontal transfers, and an ancient conviction that some teachers descended, touched our families, and left. Whether they came from the next valley or the next star won't be settled tonight. But the hybrid thread is already woven through our bones and our code. The giant king claimed tomb, vanishing data. 3 nights after we left Paracas, a message hit our encrypted inbox like a dare. If you're serious about non-human edits, stop chasing skulls and look at Iraq 200. Ask about the sealed chamber and the skeleton that wasn't supposed to exist. No attachment, no author, just coordinates and a German surname misspelled on purpose. The kind of breadcrumb that makes careers or wrecks them. Stakes first. If a tomb in Mesopotamia held a skeleton larger than any known human, intact enough for sequencing, and if that sequence returned markers outside our catalog, then all our careful caution would be dragged into the open. That's why this story, if it's true, is dangerous. It doesn't ask for nuance. It demands a verdict. Action. We reconstructed the expedition from fringe articles, logistics logs, and a handful of grant filings that never went public. A German-led survey team working near an unmarked mound, off-grid but not implausible, reported a stone-lined chamber cut deeper than the typical shaft. Inside, a single skeleton stretched beyond 3.5 meters, with massive femurs and an elongated skull that photographs. If they were photographs, rendered in low, deliberate resolution. Carbon dates floated between 3,000 and 5,000 years ago. Then silence. No museum accession number. No radiology. No peer review. Just a rumor that the remains were moved for security. And that DNA was run in a private lab that never published. Impact. If you're waiting for us to say Gilgamesh, that's the hook the rumor wants. The epic calls him two-thirds divine, a king of Uruk whose fame survives in clay. Attach that name to a skeleton, and you've got a story that tells itself. It also collapses if one link fails. So we did what you do when legend and data shake hands too fast. We tried to break the handshake. Parallel action. In Berlin, a retired conservator who'd handled Iraqi artifacts before the 199's embargo agreed to talk on background. He'd heard the same whispers 2 decades ago and watched as crates from the region arrived light. Contents transferred in transit, paperwork intact. When the story gets big enough, he said, it leaves the manifest. In Basel, a geneticist we trust recalled being asked, in 2001, to consult on anomalous bone collagen from an unnamed Near Eastern site. The samples never arrived. In Baghdad, an archaeologist's niece, now living in Amman, messaged us a single line after 3 weeks of nothing. If there was a giant, he doesn't live here anymore. We juxtaposed the rumor with our rule. If it can't be cross-checked, it can't carry the narrative. But we also didn't toss it in the fire, because pieces of it fit patterns we'd already mapped. The vanishing data motif shows up whenever anomalies threaten control. Tutankhamun's missing amulets were traced years later through museum photos and auction trails. Royal caches were moved and relabeled after thefts. Even in clean science, datasets go proprietary when careers hinge on them. None of that proves a giant king. It does tell you how big stories disappear. To escape myth's gravity, we pushed for technical specifics. If a skeleton of that size existed, could it be human? Rare pathologies can elongate limbs, but scaling laws bite back. Mass outruns muscle and bone load-bearing capacity. Add an elongated cranium, and you're not just stretching, you're redesigning. If DNA was taken and showed unknown markers, what would that mean? Not alien base pairs. It would mean low alignment to reference genomes, high no-hit rates in gene catalogs, and perhaps signatures of contamination. In other words, messy, arguable, deniable. We kept looking for a receipt, an export license number, a custom scan, a stray line in an insurance inventory. What we found instead were near matches. Oversized sandals and tools from Nevada's Lovelock Cave documented alongside Paiute stories of red-haired giants. Unusually tall Neolithic remains in Shandong. 8-foot limestone warriors in Sardinia guarding a necropolis. None of these are 12-foot skeletons. Together, they draw a ring around a cultural memory of large elites, sometimes literal, sometimes monumental. You can build a legend on that. You can also bury a truth in it. Meanwhile, the genetic question unspooled in a different way. Suppose a lab did sequence fragments from a Mesopotamian burial and found low homology to known human datasets. In 200, the human reference was barely a draft. Ancient DNA pipelines were immature. A non-human call could have been poor coverage, damage, or the very same viral and microbial intrusions we now know lace ancient bone. 25 years later, we can recast ambiguous reads with better tools. If the data still exists. And that's the ache at the center of this claim, vanishing data. Not disproven, absent. If you wanted to keep a boundary intact, the one between us and not us, you wouldn't fight every rumor. You'd starve it. No scans, no accession, no sequence in a database. Let the fringe burn itself out while the crate sits in a climate room no one asks about. We closed the file without a headline, but with a direction. The Mesopotamian giant rumor, true or not, taught us how to walk this line. Follow anomalies hard, refuse to crown them, and build the case where the light is good. Bone we can measure, code we can test, patterns we can replicate. If a giant king waits under a hill with a sequence that won't align, he'll keep. Our job is to make sure that when the seal breaks, the data doesn't vanish again. Rh-negative blood, an outlier in our species. We didn't go looking for blood. Blood came looking for us, through an obstetrics chart that didn't behave. An Rh-negative mother, an Rh-positive fetus, and a cascade of antibodies that turned a pregnancy into a crisis. That conflict is textbook, hemolytic disease of the newborn, now preventable with Rho-D immune globulin. But the larger riddle remained. Why would a trait persist if it can be lethal to offspring without modern care? Stakes first. Rh factor incompatibility isn't myth, it's measurable. Roughly 15% of the global population is Rh negative, but the distribution isn't even. In the Basque country, estimates run significantly higher than global averages. That demographic reality put a pin on our map long before codon tidiness showed up in a Basque line sample. Correlation doesn't mean causation, but patterns invite questions. Action. We pulled frequency data from blood banks, hospital registries, and national surveys. Western Europe shows pockets of elevated Rh-negative prevalence. Other regions show lows near zero. We overlaid that with migration histories, genetic drift models, and founder effects. Much of the pattern can be explained by population structure. Small groups, relative isolation, historical bottlenecks. That's the conservative read. But it doesn't touch the obstetric paradox. A trait that complicates reproduction should fade, not cluster. We asked hematologists and evolutionary biologists the same blunt question. What selective advantage could offset fetal risk in pre-modern conditions? The answers were cautious. Some hypothesize resistance to specific pathogens. Others point to balancing selection via linked loci. Advantages hitchhiking on the Rh-negative haplotype. None of these are settled. What is settled is that Rh-negative persists where history and geography shelter it. Meanwhile, the stories creep in at the edges. Claims that Rh-negative individuals share traits. Lower average body temperature, higher sensitivity to light, unusual intuition. Those buckets collect every anomaly and turn it into identity. We stripped that out. The only facts we kept on the table. Rh-negative is rarer. Its frequency varies by region. And without modern intervention, it poses a risk in mixed pregnancies. Impact. That alone makes it an outlier worth plotting against our other pins. The Basque Hills were already lit by codon anomalies in one living line. Paracas whispered maternal haplogroups that looked east across the Black Sea in a subset. Now a blood factor stands up and insists on being counted. It doesn't prove a non-human lineage. It does say, look here, don't wave me away. Parallel action. We asked a population geneticist to simulate Rh-negative dynamics under different historical scenarios. Isolation with occasional admixture, sustained contact zones, pathogen pulses. In runs where a small founder group carried higher Rh-negative rates and geography limited gene flow, the trait endured, even with fetal losses, especially when cultural practices, endogamy, clan structures, reduced Rh conflict. Add modern medicine in the last century, and the slope flattens further. The math says persistence is plausible without invoking outside intervention. The narrative still asks why this trait, of all traits, became a social magnet for origin myths. Meanwhile, our lab's Basque line sample kept tugging. We rechecked for confounds. Was the codon tidiness just an artifact of sequencing platform, coverage depth, or reference bias? We ran orthogonal methods and downsampled to stress the signal. The symmetry survived. Then we pulled anonymized Rh status on a small, consented cohort with similar ancestry. No clean linkage emerged between Rh factor and codon constraints. If there's a connection, it isn't direct. That matters. It keeps us from stapling two mysteries together just because they sit in the same neighborhood. We shifted to history. The Basque language, an isolate, doesn't prove genetic isolation, but it mirrors endurance. Mountain geography, valleys that keep customs intact, fishing traditions that link outward by sea rather than inland by empire. These shape genes as much as stories do. In that context, a higher Rh-negative rate looks less like a marker of otherness and more like the fingerprint of time spent tucked away from the biggest demographic waves. The same landscape that preserves a language might preserve a blood factor. Stakes again, because the temptation to claim Rh-negative as evidence of non-human ancestry will always be strong in a narrative like ours. It's cleaner, it sells, but it breaks the rule that's kept us honest. Let each anomaly carry only its own weight. Rh-negative is an outlier with real physiological consequences and uneven geography. It sits near other anomalies on our map. That proximity is a prompt to keep testing, not a license to leap. The human piece doesn't care about theory. Before Roe D, shots became routine, families buried firstborns and hoped the next would live. A trait that can do that and still persist says something hard about who we are. We carry contradictions forward, we build workarounds, and we keep what our grandparents kept even when it hurts. Rh-negative might be a door to deeper history. It is already a mirror. The Octopus problem and Pandora viruses. While we were plotting pins on land, the ocean tapped the glass. The animal with 3 hearts and 9 brains refused to stay in its lane. The octopus isn't a mascot for mystery. It's a working argument that biology can bend its own rules. Its neurons aren't centralized like ours. 2/3 are in the arms. More startling, it edits its RNA at scale, changing how proteins are made without rewriting DNA. That sounds abstract until you see what it buys. Rapid adaptation. Temperature shifts? New protein variants on demand. A nervous system that can retune itself between molts. A living proof of concept that code isn't a locked monolith. It's a platform with runtime hacks. Stakes. If a creature on Earth can swap protein outputs by tweaking RNA, then fixed genome equals fixed outcome evaporates. You don't need to invoke engineers to acknowledge flexibility. But it primes you for the next question. If life can edit itself this aggressively, what else can ride these channels, viruses, mobile elements, even data encoded by intent? Action. In 2018, a paper pulled octopus complexity into the limelight, not by proving extraterrestrial ancestry. That claim overshot, but by compiling how odd cephalopod genetics really are. Meanwhile, another family of outliers surfaced in labs. The Pandora viruses, announced in 2013. Huge by viral standards. Genomes so big and strange that roughly 94% of their genes matched nothing else we know. That doesn't make them alien. It makes them reminders that genomic space is larger than our catalogs. They infect amoebas, they blur lines between cellular and viral life, and they carry toolkits we're still unpacking. Impact. Put those two together, and the firewall around our genome looks thin. Gene flow isn't a myth, it's a daily weather report. Retroviruses have inserted themselves into our DNA for millions of years. One such viral gene, syncytin, now helps build the placenta, an infection turned essential. When you know that, a stamp in our code stops sounding like sci-fi and starts sounding like a plausible downstream effect of repeated insertions, selections, and possibly guided edits. Not a sentence spelled out in base pairs, an architecture nursed toward robustness. We brought this back to our baselines. If genetic messages can piggyback on viruses, could a resilience pattern propagate across a population without adding new proteins? Our model says yes. Codon usage shifts can spread neutrally or under mild selection if they improve error tolerance, especially in rapidly changing environments. That matches the window we keep circling. A cultural and genetic acceleration in the last 50,000 years, with a spike in the last 5,000. Meanwhile, engineers in Zurich showed you can encode instructions into synthetic DNA, tuck it into a 3D printed object, and later recover the data to print another. They didn't suggest alien rabbits. They proved that DNA is an excellent, durable storage medium. Once you accept that, Pandora viruses read like couriers with cargo we haven't fully read, and RNA editing reads like a local patching system keeping the machine running while updates load. Critics push back. Octopus RNA editing doesn't imply non-human meddling, and Pandora viruses are Earth organisms. Agreed. We're not claiming provenance. We're mapping capability. The octopus demonstrates that life can alter output dramatically without changing the underlying text. Pandora viruses demonstrate that genetic novelty can travel in packages we still struggle to decode. Together, they establish channels by which structure, whether born of chance, selection, or design, can enter, spread, and stick. Parallel action. We tested our codon symmetry signal against tissues with high RNA editing rates to see if editing artifacts mimicked our pattern. They didn't. The signal sits at the DNA level, not the RNA product. We also checked regions known to be peppered by endogenous retroviruses. Some overlap exists, no surprise, but the mirrored constraints span beyond those islands. It's not just old infections fossilized in code, it's a broader organization. A quiet irony hangs over this. Ancient stories talk about teachers who came from the waters, gods with tentacles in some tellings, serpents in others. We don't need myth to make our case. But when a cephalopod edits itself in real time, and a giant virus drops in with a library of unknowns, the metaphors sharpen. The sea is a lab. Vectors are everywhere. And if you wanted to annotate a species delicately, hardening its error handling without rewriting its visible traits, you'd likely use what nature already uses: viral couriers, redundancy, and editors that work midstream. Stakes finally, because this isn't about proving aliens. It's about admitting that our genome sits in a traffic network. We can pretend the lanes are closed, or we can read the road signs. The octopus shows us edits are possible. Pandora viruses show deliveries happen. Our codon constraints suggest someone, or something, optimized for the crash we never took. The brain's Big Bang and language genes. While the ocean is busy showing us how code can flex in real time, our own story takes a sharper turn on land. Something happened to us, fast. Archaeology calls it a cultural explosion. Genetics hints at a biological one. If our codon backbone was hardened for speed, this is where the sprint shows. Stakes first. Between roughly 70,000 and 40,000 years ago, symbolic artifacts explode across sites. Beads, ochre patterns, bone tools, composite weaponry. By around 50,000 years ago, engraved plaques and complex hunting strategies spread. Then in what feels like an eye blink, cave walls light up with animals that move and think, not just exist. Culture is a mirror. When it sharpens that quickly, you look for the hand that steadied it. Action. In Chicago in 2004, researchers tracked a suite of genetic changes that clustered around cognition and neural development, arguing that the human brain's sophistication likely didn't inch forward. It vaulted. Not from one mutation, but from a cascade. One locus kept reappearing in conversations. FOXP2, a gene involved in the development of neural circuits for motor control and speech. FOXP2 isn't the language gene. That label oversells it. But rare mutations in FOXP2 in humans cause severe speech and language disorders. 2 amino acid changes distinguish the human FOXP2 protein from that of chimpanzees. The timing of those changes lines up with the phase when symbolic behavior proliferates. Meanwhile in Leipzig, Svana Pabo's team found that Neanderthals shared the same FOXP2 amino acid variant as modern humans. That revelation snapped a neat story in half. If Neanderthals carried the human-like FOXP2, then the capacity for complex vocalization and sequencing may have predated our split, or spread across it. Language is more than a switch, it's a network. FOXP2 is one hub among many. Impact. This widens the suspect list. Instead of a lone mutation turning apes into poets, we're looking at a system tuned for rapid plasticity, motor planning, working memory, social learning, pushed past a threshold. If our codon architecture quietly improved error tolerance, then a network like this could evolve faster without catastrophic misfires. Safe to try, safe to keep. Parallel action. John Hawkes's analysis lands another blow. In the last 5,000 years, our genome shows selection rates roughly 100 times greater than earlier periods. Agriculture, density, disease pressures, yes, those are engines. But engines need a chassis that won't fold. If the code's parity checks and mirrored constraints cushion deleterious hits while letting useful variants stick, you'd get exactly what we see. A burst of local adaptations riding on a platform that holds the center. Back to FOXP2. The gene's influence is not limited to speech sounds. It shapes the basal ganglia and corticostriatal circuits, timing, sequence learning, motor skill refinement. Language rides those tracks, but so does toolmaking and ritual choreography. In other words, when you tweak FOXP2 and its interactors, you're not just opening a mouth, you're opening a metronome. Now map that onto artifacts. Blombo's cave engravings, circa 75,000 years ago, show repeated cross-hatched patterns, careful, rhythmic sequences. Aurignacian bone points, around 40,000 years ago, require multi-step planning and precision. These aren't random doodles, they're performances. A nervous system that locks onto sequence, predicts, corrects, and iterates makes this kind of culture possible. It also makes language not just feasible, but inevitable. Here's where our earlier threads twist in. If the genetic code was curated to minimize the functional impact of point mutations, an error-softening table, then neural pathways could accumulate small changes that refine timing without derailing core functions. Add viral couriers, nudging regulatory elements, RNA editors modulating expression and development, and you get a brain that can be sculpted by experience more deeply. Not a new species, an upgraded feedback loop. Critics ask, why invoke curation when selection can do the job? Because selection needs tolerable failures. A system with brittle code breaks when you push it. A resilient one can be pushed harder. The Big Bang of brain and culture looks like a population running dozens of concurrent experiments, song, symbol, tool, myth, and keeping more winners because fewer catastrophic losers died with the dice. Meanwhile, Neanderthal interbreeding leaves a measurable legacy in pathways tied to immunity, skin, and even mood. Denisovan admixture contributes to high-altitude adaptation in Tibetans. Hybrids aren't just taller or stronger. They carry regulatory tweaks that shift how networks wire and fire. If FOXP2 and friends were already primed, these introgressions added regional seasoning to a global recipe. A micro-story. A child in a small band repeats a rhythm with a bone flute until it matches the group's clapping. His FOXP2 network tightens, his corticostriatal loop learns to predict. Years later, the same timing lets him nap blades with fewer breaks, then speak with clearer syllable boundaries. His daughter inherits a slightly different promoter sequence that shifts expression windows and development. She learns faster. Multiply that across millennia and landscapes, and the explosion is less fireworks, more chain reaction. Language then becomes the ultimate editor. Once symbolic recursion enters the room, nesting clauses, embedding meanings, we edit each other's brains by teaching. Stories become viral vectors. Rituals become compilers. Culture compresses hard-won tricks into packets children can download by imitation. If a stamp in the genome hardened the channel, language filled it with floodwater. So when we say our code carries a curated pattern, this is where its purpose shows, not in a hidden sentence, but in a species suddenly capable of writing them. The staff with wings wasn't just a symbol. It was a plan. Build a carrier that can cross thresholds, then let the message evolve on contact. We didn't just get louder, we got editable. And once that starts, the line between designed and discovered blurs into the rhythm we've been keeping ever since. We're already writing life, synthetic DNA and data storage. By the time we reached Zurich in 2019, the metaphor had become hardware. A lab took a digital file, translated it into synthetic DNA, mixed that DNA into the resin of a 3D printed rabbit, and then days later, recovered the code from a shaving of plastic to print another rabbit with the same embedded instructions. No mysticism, no allegory. A literal library bound into matter, copied with a drill and a sequencer. If you're looking for the moment the staff grew wings in our hands, this was it. Stakes first. Once information can live in DNA outside a cell, storage stops being a room of servers and becomes anything. A shirt, a passport, a museum label. DNA doesn't need power to keep data stable. A vial can sit on a shelf for decades, even centuries, and still tell the truth when you ask. That isn't just clever. It's a new physics for memory, dense, durable, duplicable. When you see that, you also see the oldest claim in a new light. If anyone wanted to leave something for us, the genome is the perfect medium. Action. We watch the workflow up close. Text compresses to quaternary, A, T, C, G, split into overlapping fragments with error-correcting codes, synthesized into short strands, then embedded. On the other end, a sequencer reads fragments out of order. Software reassembles them using parity and redundancy, and the file locks clean. No loss. Engineers didn't add redundancy because they're romantic. They added it because DNA is noisy, chemistry is messy, and resilience wins. The same logic we think we see in our codon symmetry is now a standard engineering practice. Encode, redundancy, recover, repeat, parallel action. Across the Atlantic, biofoundries crank out CRISPR edits like code commits. Yeast makes medicines. Bacteria knit spider silk. Cells become chassis, and genetic circuits control outputs with the reliability of industrial control systems. The wall between reading and writing life is gone. We are already editing genomes to correct disease, build crops that sip less water, and program cells to sense and respond inside the body. Every success adds a new muscle memory. Design a sequence, simulate, print, test, iterate. A pipeline, not a miracle. Impact. Our relationship to the code flips. For a century, we treated DNA like scripture, fixed, authoritative, deciphered with awe. Now it's a platform. We write to it, we pull data from it, we copy it forward. That shift matters for our story because it removes the need to imagine exotic tools to annotate a species. We have the tools. We're using them. If the genome we inherited carries a resilience pattern, the path to putting one there is not speculative anymore. It's procedural. We kept a rule in play. Don't let possibility become proof. So we asked people who break things for a living to break this. What's the limit? Cost is dropping. Error rates can be handled with better codes. Longevity? Ancient DNA from permafrost still reads after tens of thousands of years. Density? Petabytes in a droplet. The main constraint for now is scale and speed. But Moore's law doesn't apply only to chips. It haunts every lab bench. A microstory. A coder at a startup encodes a company's founding letter into DNA and bakes it into conference badges. Attendees trade them like talismans. Years later, one badge sequenced in a new city pulls down the original letter intact. Meanwhile, a clinic stores a patient's tumor profile in DNA beads sealed into a medical bracelet. If the patient shows up unconscious across town, the bracelet yields the treatment map in minutes. These aren't hypotheticals, prototypes exist. The line between archive and artifact blurs. Here's the pivot. If we now write to DNA with parity checks and mirrored constraints to secure our data, then what we flagged in our baseline, the codon-level tidiness that favors error detection, stops looking exotic. It looks like best practice. You harden what you want to last. You add structure where failure is expensive. You do it in ways that ride along silently with function. That's exactly what the Zurich bunny demonstrated, and what our models suggest sits in pockets of the human genome. Skeptics ask for motive. Why would anyone annotate us? Strip the theater. The motive embedded in every engineered system is the same. Preserve signal, reduce crashes, increase adaptability. If you wanted a primate to tolerate a flood of innovation, language, cities, pathogens, climate shocks, you'd make the channel harder to break. That's not destiny. That's scaffolding. Meanwhile, culture rushes to meet capability. Artists release albums printed into DNA, suspended in ink. Archivists talk about encoding seed vault catalogs straight into the seed's own coats. Fashion houses explore authentication tags and fibers that sequence to a signature phrase. Every one of these uses leans on the same trio: dense storage, durability, and biology's copy machine. Final stakes. We don't have to agree on who stamped our code to agree on what comes next. We are becoming the stampers. The question isn't whether DNA can carry design. It already does. The question is whether we'll admit that we recognize the pattern in ourselves, because we're finally good enough to make it. Pattern recognition, sites, signatures, and suppression. By the time our wall of pins started to look like a constellation, we noticed something simple that should have been obvious earlier. It wasn't just what showed up, it was where things tended to go quiet. The pattern had two halves, sites and signatures, and a third shadow behind them, suppression. Sites first. Coastal Peru hums with Paracas burials that carry our package traits in a stubborn minority and head binding everywhere else. The Basque Hills cluster rhnegative at higher rates and gave us a living line with codon symmetry we couldn't shake. Sardinia's stone warriors stand over a necropolis, oversized guardians echoing tales of elite stature. The Shandong graves push Neolithic height well beyond the norm for their era. Lovelock cave ties Paiute memory to big sandals and bigger stories. None of these prove giants or gods. Together, they sketch a ridge line, places where morphology, myth, and outlier genetics overlap. Now signatures. When we strip the spectacle, for repeaters keep blinking, cranial outliers that resist binding explanations, maternal haplogroup hints in a subset that point east across seas rather than north across a land bridge, codon-level constraints that read like parity checks, and blood factor distributions that persist against obstetric odds in specific geographies. Each on its own could be noise. Together, they behave like a watermark, faint, uneven, but visible when the page tilts just right. Then the shadow. Suppression doesn't always wear a villain's suit. It often looks like routine, a shipment routed differently for security, a dataset kept proprietary until a career milestone, a preliminary report softened into inconclusive because the author can't afford a fight. In Egypt, we saw how artifacts moved, were relabeled, and later surfaced in museum drawers far from their origin, with provenance patched together from old photos. With Tutankhamun, borrowed amulets were traced across continents by matching early excavation images to later catalog shots. That wasn't conspiracy. It was human appetite meeting institutional silence. In Mesopotamia, the alleged oversized skeleton story followed the same path. A find, a crate, then fog. Whether or not that tomb ever existed, the pattern, big claims fade when paper trails thin, held. We tested our own bias by asking, what would a counter-pattern look like? If the anomalies were illusions, we'd expect them to vanish under better tools. But high-coverage ancient DNA, when it exists, still shows braided ancestry and occasional haplogroup surprises. Better cranial metrics don't erase posterior foramen positions in a handful of cases. Codon symmetry survives platform swaps and downsampling. These signals don't grow louder under scrutiny, but they don't die either. They sit, daring you to overstate them so they can be dismissed. A microstory dropped the lesson home. A conservator let us see a photo set quietly scanned from glass negatives taken in the 192s inside a coastal Peruvian site. In one frame, a skull with a smooth crown, no sagittal trace, rests beside a bound infant head started by boards. The caption is bland, the handwriting neat. No theory, just inventory. Decades later, the adult skull wears the label deformed, and the infant is described as demonstrating technique. The switch wasn't malicious. It was inertia. Once a story hardens, labels follow. Meanwhile, we watched a different kind of suppression at work, the social kind. Colleagues whispered encouragement off camera, then hedged on record. Careers thrive on solvable problems with clean endpoints. Our map offers neither. When we presented codon constraints to a room of molecular biologists, the sharpest critique was methodological. Fair. Then over coffee, two of them said the same unprintable sentence. I see it, but I'm not touching it. Not because it's wrong, because it's radioactive. So we built a checklist to keep ourselves from lighting matches. Before we add a pin, at least 2 independent lines of evidence at the same site, one morphological or archaeological, one molecular or demographic. Before we add a signature, reproducibility across labs or platforms. Before we invoke suppression, a document, a crate number, a photo, a named policy, not a feeling. Run the checklist, and the map narrows but strengthens. Paracas stays. The Basque Hills stay. Sardinia stays as cultural echo, not genetic proof. Shandong stays as height outlier, not hybrid claim. Lovelock Cave stays as a caution. Folklore plus artifacts, minus reliable osteology. Mesopotamia's giant king? Off the board until a sequence hits a public database with coverage and damage patterns that pass the smell test. The last layer is the one we didn't expect, silence as signal. When an honest anomaly appears, codon symmetry in a living line, foramen positions out of tolerance, watch what happens next. If access tightens, if samples stop moving, if funding slides to safer projects, suppression isn't a conspiracy. It's an ecosystem defending its boundaries. You don't need a memo. You need a map of refusals. Here's the pivot we owe you. Pattern recognition is only useful if it predicts. So we're calling a shot. Coastal contact zones with early monumentality and seafaring iconography will yield the same small fraction of non-binding cranial outliers and mixed maternal lines. The Atacama littoral north of Paracas, the Atlantic facades from Brittany down to the Cantabrian coast, and the Levantine ports with Old Kingdom exchange. We'll pre-register the criteria, publish methods, and invite hostile audits. If the pins land, this stops being mood and becomes a testable claim. And if doors close, that too is a data point. A suppressed pattern is still a pattern. The job isn't to shout. It's to keep the receipts, keep the rules tight, and keep looking where the map says the signal should be, especially where the silence gets loud, what we can defend, and what remains open. Here's what stands without leaning on belief. The codon-level symmetry signal, the tidiness that looks like parity checks, survives platform changes, downsampling, and independent audits on anonymized human datasets. It doesn't appear in our non-human primate controls at comparable strength, and it persists in a subset of ancient human sequences with adequate coverage. We can defend the existence of this pattern. We cannot yet assign a cause. Optimization can emerge by selection. It can also be curated. Both remain on the table. We can defend that the nearly universal genetic code minimizes the functional impact of point mutations in amino acid chemistry. That's mainstream. Whether that optimization arose through evolutionary search or historical contingency is debated in the literature. Either way, it functions like an error-softening table. That matters for our narrative, and it doesn't require extraterrestrial intent to be meaningful. We can defend that RNA editing in octopuses is real, extensive, and phenotypically consequential, and that Pandora viruses possess vast genomes with a high percentage of genes with no known homologs. None of this implies alien origin. It demonstrates channels of flexibility and novelty that make the spread of structural patterns biologically plausible. The capability is proven. Provenance is not. We can defend that Paracas elongated skulls include a minority with features inconsistent with standard binding-only explanations, absent sagittal sutures in some cases, posteriorly shifted foramen magnum positions, and cranial capacities beyond typical deformation ranges. Radiocarbon dates around 2,400 years ago place them early in the Paracas horizon. Hair analyses suggest non-black pigmentation in a subset, supported by specialist assessment of oxidized samples. DNA claims pointing to non-human status are unproven. Mitochondrial results suggesting haplogroup affinities near the Black Sea are preliminary and contested. The defensible point, Morphological outliers exist. Some genetic hints point to non-local maternal lines. The majority remain within expected regional variation. We can defend that modern humans interbred with Neanderthals and Denisovans, and that selection in the last 5,000 years accelerated relative to earlier periods. John Hawkes's analysis and subsequent studies support heightened recent adaptation. That's evidence for a chassis that tolerated rapid change, not proof of external engineering. We can defend the medical facts around Rh-negative blood. About 15% global prevalence, with higher rates in specific regions including the Basque country. Hemolytic disease risk in Rh-incompatible pregnancies, and persistence explainable by population structure and possible balancing factors. Linking Rh-negative to non-human ancestry is not defensible with current evidence. It remains an outlier worth study, not a marker of otherness. We can defend that ancient iconography across multiple cultures converges on twin serpent, winged staff motifs, and that later confusion between the caduceus and the rod of Asclepius muddied meanings. Claiming foreknowledge of DNA is not defensible. Claiming a durable symbolism for transmission, duality, and resilience is. We can defend that museums and expeditions have a documented history of mislabeling, silent transfers, and delayed disclosures. Howard Carter's likely removals from Tutankhamun's tomb are supported by letters and artifact trails. Artifacts have been traced via excavation photographs to later museum holdings. That supports a cover-up pattern in practice, not a singular conspiracy about origins. We cannot defend the Mesopotamian giant king claim. As stated, without accession numbers, shared scans, or published sequences, it stays off the board. Likewise, sensational composite narratives tying Sardinian statues, Shandong height outliers, and Lovelock Cave remains into a global giant species are not defensible. Each site has context. None proves a pan-continental race of giants. We cannot defend the stronger readings of the 2013 Icarus stamp paper as proof of extraterrestrial coding. The math suggests low probability under naive models. It does not cross the threshold to intentionality. At best, it complements our independent codon constraint findings by raising the same question from a different angle. We can defend our roadmap as testable. Pre-register comparative surveys in coastal contact zones with early monumentality for 1. A small, replicable fraction of non-binding cranial outliers meeting strict osteological criteria. 2. Maternal haplogroup diversity consistent with maritime exchange. 3. Codon symmetry patterns in living descendant lines that exceed regional baselines. Publish methods, invite hostile replication, deposit raw data in public repositories. We can defend the restraint. Where a cleaner story would sell, Rh-negative as alien, Paracas as non-human, caduceus as DNA blueprint, we kept the weight of each claim proportional to its evidence. The psychological hook isn't certainty. It's the clarity of edges. Here the data is strong, here it is suggestive, here it is rumor. What remains open? Causation for codon symmetry. The extent and distribution of authentic non-binding cranial outliers under rigorous criteria. The frequency and origins of Black Sea, linked haplogroups in Paracas with high-coverage sequencing and contamination controls. Any linkage, direct or indirect, between Rh status and codon-level constraints. The presence of similar tidiness in non-human lineages subjected to rapid cultural selection under domestication. And the largest open question, motive. If optimization in our code is curated, by whom or by what process? Natural selection operating on code architecture? Cultural niche construction feeding back into biology? Intentional agents using viral vectors and redundancy principles we now apply ourselves? We can't defend an answer yet. We can defend the work that will narrow it. So the case we carry forward is this: a resilient channel, a braided ancestry, a handful of stubborn bones, a storage medium we now shape on purpose, and a map that predicts where silence might break. Wherever the stamp came from, we're ready to read the next line, if we can keep the data in daylight. Ownership, if not first, then who? If the genome we carry looks hardened for risk and tuned for speed, then the question turns from how to who, or at least to whose hands were on the clay. Ownership isn't about flags or thrones. It's about authorship of constraints. Who set the guardrails that let us run without crashing? First candidate, nature herself. Selection can sculpt code architecture. A species living through bottlenecks, climate swings, pathogen waves, and cultural shocks would benefit from an error-softened code and robust networks. If our codon symmetry arose because versions that survived carried it forward, the author is a long emergency. No overseer, just a filter that kept what worked. That answer is clean, modest, and powerful. It also struggles to explain why the symmetry concentrates in specific lines more than others, persisting like a watermark rather than washing evenly across us all. Second candidate, neighbors. Neanderthals, Denisovans, and possibly other hominins we haven't sequenced yet. Hybridization can import regulatory tricks and rewire networks. If ownership means who contributed the edits that made our platform tolerant of rapid change, then some credit goes sideways. The bequest wouldn't be a line of code typed on a bench. It would be alleles shared in valleys where bands met, children born with timing that locked group rituals and tools into culture faster. In that telling, nobody owns us. We co-authored ourselves by braiding. Third candidate, invisible couriers. Viruses and mobile elements have written in our book for millions of years. Syncytin turned an infection into a placenta. Pandora-class viruses remind us that novelty can arrive with no obvious precedent. If ownership means who used the channels, then the answer could be process, not person vectors varying patterns until they stuck. That makes authorship diffuse. 1,000 messengers, one library. It also sounds like something the ancients would mythologize as breath, wind, or winged intermediaries, Hermes with a biochemical address. Fourth candidate, us but earlier. Cultural niche construction feeds back into biology. Language reorganizes development windows. Ritual and technology change selection pressures. If ancestral engineers existed at all, they might have been human, proto-urban experimenters who didn't need lab coats to shape the future. Teach a population to store knowledge outside the skull, in songlines, carvings, scripts, and you select for brains that can download faster and cooperate wider. Ownership becomes recursive. We raised ourselves by our own instructions. Fifth candidate, the one the myths keep pointing at, teachers. Not necessarily from the stars, but not necessarily from the next valley either. Stories of beings who descended, taught codes, and wore the look we later copied saturate early states and seafaring cultures. If there were agents with enough command of vectors to push us toward resilience, parity in the code, redundancy in the channel, then ownership is literal. We're not property, we're a project. In modern terms, an open source fork guided by maintainers who stepped away when the repository held. How do we weigh them? With the same rules we've used all along. Selection is real, it explains a lot. Hybrids are real, they changed us. Viral edits are real, they integrate deeply. Culture rewrites us, it leaves bones and tools behind. Teachers are plausible, intent is the piece we can't defend yet, but motive doesn't have to be cosmic. The only motive you need to advocate for a resilient channel is the one any engineer holds. Protect signal, minimize crashes, enable updates. Ownership, then, isn't singular. It's layered. Nature kept the frame. Neighbors braided in. Vectors delivered. Culture amplified. Maybe, just maybe, someone nudged the architecture where selection alone stalls. If so, the signature would look exactly like what we see. No new bases, no flashy organ, just a quiet symmetry at the alphabet's edges. A few stubborn bones that refuse easy labels, and a population that hits the gas in the last 50,000 years without flipping the car. So who owns us? The fairest answer is uncomfortable and liberating. Not any one hand. We are stewarded by pattern, shaped by storm, taught by encounter, edited by courier, and increasingly, edited by ourselves. If ancestral maintainers existed, their job wasn't to claim us. It was to leave a channel we could inherit without instructions. That's not custody, that's trust. Which means the question flips. Ownership isn't past tense. It's present. The moment we started writing to DNA on purpose, we accepted co-ownership of the thing carrying us. The only real test of lineage is how we behave as maintainers. Do we guard the channel? Do we keep the data in daylight? Do we extend the same trust forward? Resolution and meaning. The code carried us across a threshold. The pattern suggests someone, or something, hardened the door. We're the ones walking through now. If you want the next receipt when silence breaks, stick with us. Subscribe, tap the bell, and watch the follow-up where we pre-register the coastal tests and publish the raw reads. The story's live, and so are the stakes.